Let me state at the outset that Mendel's experiments on plant hybridization are, in my view, and I've stated this in print and publicly elsewhere, the best experiments ever done. I'll go even further and state that they are a work of genius, and everyone who is interested in science should read them. What they did, here's Gregor Mendel, every talk must begin with a picture of Mendel. What Mendel's experiments did was they suggested the laws of segregation and independent assortment, which are the basis of modern genetics, and provided strong evidence for those laws. The former law states that variation from contrasting traits is associated with a pair of factors which segregate to individual reproductive cells. The latter states that two or more of these factor pairs assort independently to individual reproductive cells, and the factors, which we now call genes, are dominant or recessive. Now, when I first started working on Mendel's experiments, after all, I am a professor of physics, my only knowledge of Mendel's experiments came from a paragraph or two in a high school genetics, in a high school biology text, and one lecture on it. So I will provide a refresher course for those of you who are like me.
There was an Austrian monk called Gregor. He looked at something never done before. I said Gregor Mendel was his name. He wondered why some things look the same. Using bees, he looked to their height. He worked all day and all night. The first G cross was a tall and a dwarf one. Their offspring were all so equal. He was at no loss. He went for a second cross. He did it with two of these tall. What did he see? I said, what did he see? F2, one's wrong. Tall ones there with three. Now, what was his conclusion? To him, there was no confusion. Features came in hereditary units. This was his message. One was dominant and the other recessive. His work turned out to be quite pathetic. It was the start of modern genetics. Word to the brother. Peace.
The video actually gets Mendel's basic genetics right. If you cross two hybrids, you will get three that are dominant and one that's recessive. You can see it here. You get three. Now, Mendel conducted his experiments over 8 years. He spent 2 years working on finding varieties of peas which indeed bred true, that they showed, and he picked characters that showed no blending. And indeed, in his first set of experiments, he crossed a pure dominant with a pure recessive. For example, in color of the cotyledons, he crossed yellow with green and obtained all yellow. His results in the second cross, where he crossed the hybrids, were the following. He got, you'll notice, the expected ratio of 3 to 1, which was indeed what his model predicted. But one of the things that he worried about was that in the dominant, 2 of them would be hybrid and 1 would be pure dominant. And so he designed an experiment in which he would check whether he got 2 heterozygous plants for 1 homozygous. And what he did was he planted 100 of these dominant plants and sowed or cultivated, the German word is not easily translatable, there's some dispute over it, and looked at the offspring. If all 10 were dominant, he classified it as homozygous. If a recessive showed up, he classified it as heterozygous. There are some probability problems with that, but I won't go into that here. And here's his results of just some of the plants. He recognized that there was wide variation in individual plants. This is the 3, should be 3 to 1. You'll notice he gives varying results. He's not averse to that. He found that in the first experiment, he actually once found 43 round, 2 angular, and 14 round and 15 angular. In this experiment on the color of the cotyledon, he once found 32 yellow, 1 green, but also 1 with 20 yellow and 19. His F2 results are clearly, he used the seed characters because he got many more results. Seeds are also in the first generation. You can see them more easily. And he gets clearly 2 to 1, except for his experiment on pod color, where he got 60-40 and 1.5 is the ratio. He redid the experiment and got 65 to 35, which he thought was much closer to what he expected. And he said, accordingly, the average 2 to 1 seems insured. A modern statistician would say that actually 60-40 is within 1 standard deviation of what the expected results should be. Now, he also worried about whether or not, if you had 2 character features that you looked at, whether they would transmit independently. And he again works on double hybrid, and his expected results are, when he crosses them, are 9, 3, 3, 1. You should get round and yellow, the 2 dominants in a ratio of 9. And with 1 recessive, you should get 3. And with 2 recessives, 1. And indeed, he finds these are his results: 9.06, 3.11, 2.9, quite close to what his model predicts. And indeed, the number of plants he gets are in a line with, again, what his model predicts, a ratio of 1, 2 to 4 for the different genotypes. These are the phenotypes. Okay, so again, he works on it. He tries it again for 3 characteristics. These were, again, the 2 seeds, the color of the cotyledons and their shape, and he used seed coat color. And he finds, again, exactly what he would have expected, a ratio of 1 to 4 and 8 for the different genotypes. And so this indeed was the results that Mendel expected, and they provide very, very strong evidence for his model. Now, one of the things that Mendel did was during these experiments, he typically put the dominant in the egg and the recessive in the pollen, and he worried whether or not that would work if he reversed that, whether it made any difference which factor was in the egg or the pollen. And he designs oops.
Sorry to interrupt, Allan, for a second. We're losing you a little bit. Can I just remind everybody not to be using Wi-Fi, so that we've got a weak signal at this end, and if lots of people are using it, it can create a problem. Okay, so maybe if you could go back one slide, that would be great. Sorry.
Okay, to the trifactorial experiment, where he's using seed color, seed shape, and seed coat color, seed cotyledon color, and seed coat color. And what he predicts is that the different genotypes you get 27 different genotypes. He predicts the ratio for those which are the pure, there should be have a ratio of 1, 2, 4, and 8. The 8 being all 3 being hybrids. Now, as I said, in his experiments, he typically put the dominant in the egg and the recessive in the pollen. And the question is, did that make any difference? So Mendel designed an experiment where he crossed a pure dominant with a hybrid, and he used the egg cells from the hybrid and the pollen cells from the pure dominant. And in this, you expect genotypes to give you a ratio of 1 to 1, as they do. And then he reverses it. He uses the egg cells from the pure dominant and the pollen cells from the hybrid and gets the same thing. He also tries it with the egg cells from a hybrid and the pollen cells from a pure recessive. Sorry. And again, he predicts ratio of 1 to 1 for the genotypes. And when he reverses it with the egg cells from the pure recessive and the pollens from the hybrid, he again finds this. So this result is his last result, although he continues some of his experiments, for example, on the seed characters, color and shape. He continues those for 6 generations. These experiments lasted over a period of 8 years. Some of the other experiments he went over for 5 or 4 generations. Now, one thing that people do not necessarily, historians don't always recognize, is in fact that Mendel's garden and his experiments served yet another purpose. They provided food for the monastery, and indeed there have been comments later that perhaps using peas was not always appreciated. Now, I would want to argue here that Mendel's experiments are an almost perfect set of experiments. you can hardly imagine a better one. Experiments can be good in various ways. They can be conceptually important, methodologically good, technically good, and pedagogically important. Now, clearly, Mendel's experiments are, as I've shown, conceptually important. The laws of segregation and independent assortment, and the idea of dominant and recessive traits are the foundations of modern genetics. Now, Mendel provides evidence for that, and indeed, his evidence is overwhelming. The probability of getting a worse fit to his model is 7 times 10 to the minus 5. This becomes an issue later after Fisher calculates this. Now, his experiments actually were exploratory. At the time, not only was there no good data on plant hybridization, there was in fact no model. And in fact, Mendel not only provided the data, but he provided the model. Now, it was technically good in terms of his design. He quite carefully relates his results to his expected model results, and he provides statistical evidence. The methodology is just overwhelmingly good. Mendel provides a clear connection between theory and evidence. He also worried about confounding factors, for example, the question of whether you could always distinguish between yellow and green cotyledons. And he argued that, in fact, you could. And indeed, the experiments are pedagogically good. I borrowed 3 introductory genetics textbooks from a colleague in a biology department, and they each devote at least 10 pages to Mendel's experiments. But I can do no better in summing up than to quote R.A. Fisher, the distinguished geneticist and statistician, who says, Falk had overlooked in his chosen field experimental researchers, conclusive in their results, faultlessly lucid in presentation, and vital to the understanding not of one problem of current interest, but of many. Thank you.